Hello everyone, welcome to QPT. Using a type of artificial intelligence known as deep learning, MIT researchers have discovered a class of compounds that can kill a drug-resistant bacterium that causes more than 10,000 deaths in the United States every year. In a study appearing in the journal Nature, the researchers showed that these compounds could kill methicillin-resistant Staphylococcus aureus (MRSA) grown in a lab dish and in two mouse models of MRSA infection. The compounds also show very low toxicity against human cells, making them particularly good drug candidates. A key innovation of the new study is that the researchers were also able to figure out what kinds of information the deep learning model was using to make its antibiotic potency predictions. This knowledge could help researchers to design additional drugs that might work even better than the ones identified by the model. The researcher says that the insight here was that they could see what was being learned by the models to make their predictions that certain molecules would make for good antibiotics. MRSA, which infects more than 80,000 people in the United States every year, often causes skin infections or pneumonia. Severe cases can lead to sepsis, a potentially fatal bloodstream infection. Over the past several years, the MIT researchers have begun using deep learning to try to find new antibiotics. Their work has yielded potential drugs against Acinetobacter baumannii, a bacterium that is often found in hospitals, and many other drug-resistant bacteria. These compounds were identified using deep learning models that can learn to identify chemical structures that are associated with antimicrobial activity. These models then shift through millions of other compounds, generating predictions of which ones may have strong antimicrobial activity. These types of searches have proven fruitful, but one limitation to this approach is that the models are black boxes, meaning that there is no way of knowing what features the model based its predictions on. If scientists knew how the models were making their predictions, it could be easier for them to identify or design additional antibiotics. First, the researchers trained a deep learning model using substantially expanded datasets. They generated this training data by testing about 39,000 compounds for antibiotic activity against MRSA and then fed this data plus information on the chemical structures of the compounds into the model. To figure out how the model was making its predictions, the researchers adopted an algorithm known as Monte Carlo tree search, which has been used to help make other deep learning models such as AlphaGo more explainable. This search algorithm allows the model to generate not only an estimate of each molecule's antimicrobial activity, but also a prediction for which substructures of the molecule likely account for that activity. To further narrow down the pool of candidate drugs, the researchers trained 3 additional deep learning models to predict whether the compounds were toxic to 3 different types of human cells. By combining this information with the predictions of antimicrobial activity, the researchers discovered compounds that could kill microbes while having minimal adverse effects on the human body. Using this collection of models, the researchers screened about 12 million compounds, all of which are commercially available. From this collection, the models identified compounds from 5 different classes based on chemical substructures within the molecules that were predicted to be active against MRSA. The researchers purchased about 280 compounds and tested them against MRSA grown in a lab dish, allowing them to identify 2 from the same class that appeared to be very promising antibiotic candidates. In tests in 2 mouse models, one of MRSA skin infection and one of MRSA systemic infection, each of those compounds reduced the MRSA population by a factor of 10. Experiments revealed that the compounds appear to kill bacteria by disturbing their ability to maintain an electrochemical gradient across their cell membranes. This gradient is needed for many critical cell functions, including the ability to produce ATP, that is, molecules that cells use to store energy. An antibiotic candidate that the research lab discovered in 2020, halicin, appears to work by a similar mechanism, but it's specific to gram-negative bacteria, that is, bacteria with thin cell walls. MRSA is a gram-positive bacterium with thicker cell walls. The researcher says that they have pretty strong evidence that this new structural class is active against gram-positive pathogens by selectively dissipating the proton motive force in bacteria. The molecules are attacking bacterial cell membranes selectively in a way that does not incur substantial damage in human cell membranes. The researchers says that their substantially augmented deep learning approach allowed them to predict this new structural class of antibiotics and enable the finding that it is not toxic against human cells. Thanks for watching this video. As of now, you can buy my ebook Emerging Technologies for Profit from my blog for just $5. Share this offer with your social media groups. Thanks.